Hello, everyone. I welcome the students, scholars, researchers, and scientists all over the world. This is Advanced Multiple YouTube channel. Advanced Multiple Incorporation actually a knowledge processing and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as scientific executive with the Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Okay, well, today I'm going to share my knowledge, my experience, and my research under the title of Types and Introduction of Immunogenic Substances like vaccines intended to induce immunity. Okay, for this purpose, I collected the scientific research information and the data from different sources like PubMed, Medline, Scopus, Google Scholar, National Library of Medicine, MBAS, directly of Open Access Journal, ProQuest, CNKI, that is China-based database company, provides scientific research information all over the world. CNKI stands for China National Knowledge Infrastructure. Some information collected from ResearchGate and Crossref. Okay, well, before moving forward, I would like to talk a little bit about the vaccine. Like vaccines can be defined as pharmaceutical suspension or solution of an immunogenic substance or compounds that is intended to induce active immunity against antigens or the toxins of the microbial viral diseases. A vaccine is a biological preparation that provide active acquired immunity to a particular infectious disease. A vaccine typically contains an agent that represents a disease-causing microorganism and is often made from weakened or killed form of the microbes, its toxin, or one of its surface protein or component. Okay, well, this is very simple, basic, fundamental diagram that actually review the cellular and antibody mediated immunity. We know that in a human being or in normal regular human body there are the two main type of the immunity. The first one is the cellular immunity or cell mediated immunity. Second one is the antibody mediated immunity. In this cell mediated immunity, there are the cytotoxic cells involved. They produce some toxic material or the toxins called as cytokines and that kill the infectious cancerous cells. The antibody produced against particular specific antigen or the microorganism to kill or destroy the toxins. And we know that there are the two main type of the antigens. Antigen are two main type. The first one is the virus. Second one are the non-viruses. For the viruses, we have class 2 major histocompatibility complex MHC, and for all non-viruses, toxins, non-viruses, antigens, we have major histocompatibility one, type 1 and type 2, or the class 1 or class 2. Whenever the antigen, toxins, or microorganism entered into the human body, then our immune system is activated. Immune system activated means the initial step at very start of the immunological response, the antigen presenting cells are activated. They capture and they engulf the antigen. These antigen presenting cells, maybe the macrophage or maybe the dendrite cell, they capture and engulf the toxin or the antigen, process it, and present it like an epitope. It may be the viral epitope, maybe the non-viral epitope. These presented at the surface of major histocompatibility complex and then presented to the T cells, T helper cells. When T helper cells, it receive the epitopes through its TCR. TCR mean T cell receptor, then T helper cells are activated. When T helper cells are activated, they start producing the interleukin. Interleukin are three main type. There are also some other type of interleukin too, but in immunity or in immune system, we consider only three main type of the interleukin: interleukin 2, interleukin 4, and 5. Interleukin 2 actually motivate or activate the cytotoxic cell, then cytotoxic cells start producing and synthesizing some toxins, cytotoxin, that may be the histamines, maybe the cytokines. These toxic and poisonous material then destroy, damage the infectious cancerous tissues. On other hand, the interleukin 4 and interleukin 5, they start produce, they actually activate the beta cells. Beta cell then converted into the two main types. First beta cell, they become the plasma cell, and second one become the memory beta cells. The memory beta cell, they save and protect the information. Whenever same antigen came again into the body, they enter into the same person or individual. Then the memory cell, they activated, they repeat the same system and kill and destroy the antigen. The beta cell or the plasma cell, they start producing the antibody then very particular or specific for the antigen. All different kind of antigen having their own particular antibodies. So the in same way, T helper cells also converted into the memory T helper cell, and they save the data and information regarding the antigen. Okay, well, now we are going to move forward to talk a little bit about molecular structure of the antibody. Antibody having two main chain. The first one is the heavy chain, and last one the light chain. These light and heavy chain, they bind together by the disulfide bond, and a regular antibody having two part or two region. The first one is the variable region, second one is the constant region. The constant region actually stay constant. It's not changed. Its chemistry, configuration, orientation, shape, that stay constant to keep the structure in its integral regular form. And the variable region is variable. Why it is variable? Because it have to attach with the variety of the antigens. It have to kill variety of the organism, variety of the toxins. So God has given the competency to the antibody at the variable region to change its chemistry, change its orientation, change its shape and structure all the time, to capture all different kind of the antibody antigens, all different kind of the toxins, all different kind of the microorganism. Okay, well, now we are going to talk a little bit about the type of the vaccine. There are the 8 different type of the vaccine. The first one, the live attenuated vaccine. Second one is the inactivated vaccine, then conjugated vaccine, subunit vaccine, recombinant vaccines, synthetic peptide vaccine, multi-vaccine system, and vaccine used against the cancers. Okay, well, now we are going to talk a little bit one by one about all these 9 kind of the vaccines. Live attenuated vaccine, these are the live but weak vaccines. They most of the time produce the renal infection because these antibodies are excreted through the liver or through the kidney. If they are excreted through the kidney most of the time capture it. If it capture it, it may produce some kind of the complication in the kidney. So renal infection may be induced or produced by live attenuated vaccines. It is strongly attached with the antigen, so it have very strong association with the particular antigen. They alert the T cell and B cells, and they have memory cells to save the information and the data regarding the antigen. Second one is inactivated vaccine. These are the killed vaccine, microbes killed with the heat or chemical or with the ultrasound rays or X-ray rays, and they are killed, and then they are converted into some dosage form, maybe solution, suspension, emulsion. Alert only the B cells. Humoral immunity, the first dose, and protective reaction is gained after with the booster dose. Not as much effective as like a live or attenuated vaccine. Conjugated vaccine, microbes with protective capsule, polysaccharides hide antigen, link polysaccharide capsule to antigen toxoids that an immature immune system can be recognized, usually used in babies. Okay, well, the fourth one is the subunit vaccine, antigen of the microbes to stimulate response created in by two ways, by two methods, by the chemical break microbes and recombinant technology. Fifth one is recombinant vaccine. Genes from the disease caused microorganism are inserted into the vector. These vectors then injected uh, and the purified antigen. Example of such kind of the recombinant vaccine are the hepatitis B vaccine. The sixth type of the vaccine is the synthetic peptide vaccine. identify the specific region of the pathogen that are not so harmful would elicit a protect immune response of the body. This short chain of amino acids, peptides, and these represent these region are generally part of a large protein on the surface of parasite. These peptides may be prepared either by chemical, synthetic, or biological synthesis system. Multi-vaccine system is based on recombinant DNA technology. Immunogenic peptides of more than one pathogens are incorporated together into the same carrier or carrier. The carrier is not the conventional plasmid or the virus, but strains of another pathogen whose disease-causing ability have been destroyed, or they naturally may not have the ability to produce the infected. The two critical bacterial strain name BCG, that is Bacillus calmette-guerin, the attenuated strain of the tuberculosis organism implied as tuberculosis vaccine, weakened strain of pathogen Salmonella, they essentially cause the typhoid. Okay, well, the last one is the second last is the cancer vaccine deployed by the newer genetic and genic technology. The cancer research UK developed cancer vaccines that provide legitimate and adequate protection against some kind of the cancer-related disease, for example, glandular fever, nasopharyngeal cancer, and cancer of the jaw. Okay, well, in conclusion, the improvement of the existing vaccine and the discovery of new vaccines require an understanding of the immunological principle of vaccination. Great challenges remain, particularly in the term of target pathogen for future vaccine candidate and also the acceptance of vaccination. Therefore, the understanding the principle of vaccination allow development of the vaccine and the control of the mortal infectious diseases. Okay, before closing, I would like to salutate and greet to my friends, my colleague, my coworker, and these are the Shiri Bachkar, Wolfgang Bang, Daniel, Dustin Hawker, Marwa Al Mogherbi, then Robert Heyman, Unza, Noreen, Teremil, Ishtiaq, Arslan, and Amin. These are my people. They helped me a lot to make this project successful today. I also acknowledge the kind help and support of Advanced Multiple Incorporation and the Intellectual Consortium of Drug Discovery Technology Development Incorporation. Okay, well, I appreciate you for watching the video and staying with me, spending very beautiful time today. See you soon in my next video. Take care and bye for now.